Tripartite in.
Okay. Hi, Manjari.
I think she's still setting up.
Right.
Good morning.
Hi, good morning.
Yeah,
Hey, how are you?
I'm fine. Yeah, how are you all surviving? Are you all working?
Partially, we are doing a rota of 10 days amongst
Okay.
four of us, so
Uh-huh.
not much. And we are doing
Mm-hmm.
some video consults on the web.
Mm-hmm.
That's about it because there was a huge cluster of cases in the hospital, so the
Yeah.
BMC shut it down.
Okay.
Subsequently, all the residents have tested negative.
Okay.
And from today, I think they are restarting essential services, which is radiotherapy, daycare, chemo, dialysis,
Uh-huh.
and casualty.
Okay.
But the regular
OPD.
specialties, they are not going to function.
Yeah,
Maybe
and the OPDs also must be out of action.
yes, yes. So in fact, the hospital has a website on which we can do online consults.
Mm-hmm.
Reliance has the same. So
Mm.
I guess that's going to be the future for now.
Yeah.
How about you folks?
We are doing teleconsults. So we have the appointments, patients' numbers. So the files are bought in the previous day, and then we call them up from 3 designated mobile phones, which are not personal,
Right.
and we take their complaints and draw out an action plan. If there's a change of medication, we send it by SMS or WhatsApp the photo of the prescription, OPD
Right.
prescription to them. The
And how
problem
are you?
is the problem is that the staff who was getting our files and taking them out from records and all that has tested positive.
Right. And how are your residents? They are all fine.
Residents all fine, but the staff, the ground staff which was taking out the files and giving them to us, because they are positive today, there's no OPD.
Right.
Everybody is getting tested, and then because see, we can look after ourselves, but these people, they all come from very crowded backgrounds, so
True. True. No, we have the same situation in all the private hospitals because one of the elephants in the room is actually if someone tests positive and then
Mhm.
they need to be isolated and taken care of,
Mhm.
then there are lots of new overheads because you
Mhm.
need to isolate them in their own rooms. They have taken
Yeah.
hotels. So who pays for it? Obviously the
Mhm.
institution. So the money
I think
managers,
everything will have to be done in slow motion. We can't be working at the same pace as we were before.
not only that, I think the biggest thing that private
Cost.
healthcare will have to do is will have to put aside profit for the next 2 years.
Yeah, some private hospitals in Delhi have grossly cut down salaries. Like, I don't know, the hearsay is that Medanta is half the salaries of doctors to meet up with the costs. So that has also happened.
So that's going to be a disaster because if healthcare and doctors maybe will survive because they have savings, but people who
Yeah,
work in the institutions, nurses and staff, there's no way that their salaries can be truncated.
they're already low.
They're anyway just on the fringes of survival. So we can't.
I don't know. This whole thing seems to be so surreal.
And none of the new news coming in from China is very positive. So
Yeah, I don't know. No way. I don't think, I've stopped listening to news. I just don't tune into the channels. Like
right,
my own hospital is becoming news now because every day there's something. So last week there was one staff who was positive, and yesterday night there was a message that the fellow who used to come and take the files after the OPD, he's also positive. And they all are meeting in the basement. So obviously the basement people have to be quarantined now. So every day our hospital news itself is so overwhelming. Where do you have time to listen to the national or international news?
yeah, and we also live in an age of too much information. So
Yeah, I think these webinars are one way to stay sane, because
absolutely,
you are not
absolutely, and maybe one way also to evolve
yeah,
for
yeah,
all of us
yeah, because I don't foresee a physical face to face conference in the
for two
next one
years.
year. One
I
year
think
at least, maybe 2 years,
one
who
year at least.
knows.
Yeah, maybe two. Who knows?
I think this is a better way. It's more cost saving for everybody, and maybe
Yes,
the pharma can actually help hospitals meet out the expenses which you're talking about. If they're
Right.
saving on conferences, imagine the amount that the
Yeah,
industry pays.
you're not putting up staff
Yeah,
and
so
everyone
you're
at the
not making
hotel.
them
You're
travel. You're
not
not.
making them
Yeah,
travel.
so maybe some something can be drawn out at, I don't
Right.
know. There are ethics issues, but
Very big ethical issues.
somebody has
And
to.
looking at the governmental stance, I don't think they will allow
No,
anything like
no.
that
It has to be done as a philanthropic
as
donation
a
or something,
CSR.
something like that.
So, Sudhir, are we ready to start?
Once
Yeah, it's 5 minutes.
5 minutes, you can
5
open it
minutes.
up.
Yes, I'll open it up, sir.
Hi, Manjiri. Good morning.
Hi, morning. Good morning. How are you?
I'm good.
You're keeping us sane during the
I
crisis.
know I'm keeping myself sane. It's a very selfish time. Otherwise, I would
Yeah,
have gone mad.
yeah. So, somebody said, Madam, there are too many at the same time. So I said,
Uh,
think of it as a conference with parallel sessions. What do you
yeah,
think?
exactly what I thought. And you
What?
don't have to attend everything. No,
No, but
nobody.
the topics are so good that you feel like it's like a refresher course. So you feel like,
Yeah,
so
that's what
yesterday I
somebody
was
told me. I told
listening.
somebody the same
Uh,
thing. I said, you don't have to. He says, no, no, but the topics are so good, you feel
yeah.
guilty, you don't attend.
Yeah, you feel bad, that you've not like yesterday. The approach to movement, hyperkinetic movement disorder, was
Yeah,
so
super.
good, so basic
Yeah.
that for anybody who's not in movement disorder like me, it's just like
It
refreshing the
was
whole thing.
just fantastic. I
Yeah,
Yeah.
mean, it
so in
was.
fact, that's what I told Kailash. He was going to give us some. I said, no, no, we want the
No,
basic thing.
basic,
Movement
basic.
disorder group, mein you give all these high level talks. It's okay. Here we
Yeah.
want the
The only
basic
thing is,
stuff.
if we can have some kind of Google Doodle on which webinar goes on when, then I
I
think
know
we'll be
that
able to spread out some way.
we'll have to
So,
have some.
so the major societies like MDS-ICON, IES, IAN,
Yeah,
FINE, all these people, they get together and have a Google Doodle
decide.
of
Yes.
their activities, so that there is no overlap and people also feel that they're able to attend everything and not,
Yeah,
maybe
actually, you
something
know
has to
what?
be thought of.
When we started, no, that time there was nobody. Then when we started, then MDSI was already doing. So I asked them. They said, "Yeah, leave Thursday and Saturday for us. That those are the days we are doing." I said, "Fine." Then the stroke people said we'll have in the morning, so we didn't have any problem. And then Sita said she wanted to do epilepsy, so I said, "You tell me what day, and we'll not do it on." So initially we started like that. Then all these other pharma companies also joined in, and then it
Yes.
has become free for all.
Yeah, fine has been a kind of torchbearer. So you've got this Me Too phenomenon also. So,
Yeah,
but
yeah,
it's
Not me
okay. I
too.
think,
Not me too. See, because every society has to exist,
right.
and there's so many. Now,
yeah,
I mean,
yeah.
previously we didn't have movement disorder. We didn't have headache. Now we have a headache.
True.
Now we have movement disorder. Now we previously we of course had epilepsy, and
Yeah.
iron was always there. So,
Yes,
and
yes.
stroke is also there. So everybody, every
Yeah,
society
this,
has to do its bit. Like
yeah, yeah,
they
yeah.
feel
No,
they have to do their bit. I don't
no,
know.
and
Yes, fair enough.
I think now we have to realize that this is for the next 1 year
This
definitely,
is
as
the
you
way
are
to
saying.
go. This
This
is
is
the way to go.
how it
I
will
mean,
happen.
maybe longer than 1 year. Who knows? Maybe
Yeah,
longer.
Yeah,
yeah.
yeah.
So, yeah.
We'll wait another 2-3 minutes, then you can start. I'm around.
Yeah, yeah, Sudhir, I can see you.
Okay.
I think the worst is the fact that many of our households are running without house help. So that is the worst.
Yes, yes. So we are sweeping and we are cleaning the dishes and
Yeah.
it's
So
all,
it's like in one way it's like living in US. So
yes,
you have to
it's
do
like we have
everything
gone
there.
back to UK in those
Yeah.
days when we were doing all the household work. So
So
it's
that's the only downside of it. But in one way, it's good also. So we are having only 2 meals a day. We've cut down, one meal straight
excellent.
down because everything has to be done by self. So minimize.
Super. So we fortunately brought Anand back on the 18th of March before the shutdown. So luckily
Yeah.
he's here.
Where was
So
he?
he was in Indiana. He was doing computer science. So
Oh,
he's back here with us.
yeah. Some people are still stuck there.
Yeah. Okay, I think Manju,
We'll
we can
start. Yeah.
start. Yeah.
Yeah. So, good morning. I can see there are about 60 participants, and today we have a very interesting topic, which is going to be discussed by Dr. Joy Desai. And we know his deep interest in sleep, and sleep is basically a neural phenomenon. So, as he says, neurologists must be interested in this for their, own sake. And again, it's very multidisciplinary, right from basics, which is something which we are covering today, to neurologists, to respiratory, to dental, to ENT, to psychiatrists. All have sleep labs, at least in this country. So he's going to be talking to us about glial neurobiology, and that's something very interesting because we often only think of the brain as neurons, but there's lots more to these glia, oligodendrocytes, and astrocytes, and probably they have undisclosed, undetected role, which he's going to enlighten us about. Over to you, Joy.
Thank you so much. Thank you. At the outset, it's good to be back. And major disclosure, I'm not here to take a lecture. I'm actually here to share my journey as a student. And my interest in glial biology started when one of my patients who was being treated by me for epilepsy, he had a breakthrough seizure, and I admonished him in the clinic and told him that, look, just taking medication is not good enough. You need to sleep on time. And he said, no, no, I'm sleeping 8 hours, but I sleep at 1 o'clock. And when I counseled him to sleep on time, he said, but doctor, why does that happen? And that set us off on that journey to try to understand what is it that happens in the brain that alters the fine balance between neuronal circuitry. And what I'm sharing with you all today is many hours of study that transpired from that time to now. And I hope that you enjoy this journey as much as I have. So, okay, there's a paradigm shift in how we see glia. The term neuroglia originates from the Greek word for glue, and we know that glial cells are the astrocytes, the oligodendrocytes in the CNS, the Schwann cells in the peripheral nervous system, microglia, and it's now believed that maybe tancytes in the hypothalamus could also be of glial origin and may have a overlap role with glia, and satellite glial cells in the sympathetic system. And they were all perceived to be a structural scaffolding of the brain. And at least what I was taught in my neurology training is that the neurons are electrically active from their depolarization shifts of ion channels, and they are associated with neurotransmitter release at synapses. And we were taught that this is the prototypic component of functioning neural networks. Now, all these concepts changed in the mid 1990s when it became possible to label and to study calcium influx in cell studies. And lo and behold, it became very clear that glial cells also chatter just like neuronal cells. And the difference is that neuronal cells chatter due to electrical polarization shifts driven by ionic channels, whereas glial cells seem to chatter based on calcium fluxes within the glia. The fact that glia are active and they change was recognized very long ago, and this is the actual painting made by Santiago Ramon y Cajal in 1897. This is from the Madrid Museum, the Cajal Museum in Madrid. And he actually was the first to hypothesize that maybe the glia change their structural morphology during wake and sleep states. And you can see the lovely diagram where he's shown the astrocytes, and you can see the neurons and how the astrocytes embrace not only the neurons but also the vessels. And he proposed that there is a structural change in the wake and sleep state of these glia. He was perhaps right, perhaps not in the way that he meant. He actually said that, but he was the first to perceive this. And then there was a lot of seminal work, essentially in the early 1990s, which demonstrated that you could, through optogenetics and optokinetics, you could label calcium currents in astrocytes and oligodendroglia and microglia. And the labeling could reveal that the glia talk to each other via fluxes in calcium. And we began a journey of understanding what these glia do, and perhaps an expanded understanding of the roles they have to play in the CNS. It also became very clear that astrocytes outnumber the neurons in the human brain by a very large number, almost 1.4 times more astrocytes than neurons. That's the figure we have. And these astrocytes can modulate not only neurons, but they can modulate through syncytial or ensemble networks, astrocytes at a distance. And it is believed that one astrocyte may actually be communicating with almost 500,000 other astrocytes and a very, very large number of neurons. So the cardinal understanding from 2007, when Philip Hayden, we'll come back to him in the next couple of slides, demonstrated that there are some key astrocyte functions, and most of these key functions seem to occur in the sleep state with a greater alacrity and enthusiasm than in the wake state. And these three key functions are shown here in this diagram. The first is neurotransmitter clearance, and the second is vascular coupling. We know that the foot processes of the astrocytes, they closely embrace the cerebral vessels in the brain, and that has a purpose. And the third and most important is potassium buffering. And subsequent to that, three more additional roles of glial function emerged, and we will talk about it in the slides to come. So Philip Hayden, who initially worked in Pennsylvania and then moved to Tufts in Boston, is thought to be the key researcher at a cellular level who demonstrated that the astrocyte and the neurons exhibit what is known as a tripartite synapse. And this is the diagrammatic expression of the tripartite synapse. On the left, you see an electron microscopic picture of a pre and post synaptic neuron, which is shown there. And on the right, you see a diagrammatic cartoonized version of what you are seeing on the left, which shows you the presynaptic neuron is in violet, the postsynaptic neuron is in blue, and the astrocyte is shown in light green. And what it shows is that it is a tripartite synapse, and the astrocyte is regulating not only the presynaptic neuron, but also the postsynaptic neuron, and not at one site, but at more than one site. So what does it actually do? There is a close association of the astrocytic process with pre and postsynaptic terminals, and this plays a very crucial role in potassium buffering or clearing of potassium ions that accumulate following neuronal activity. The second key contribution is the uptake of synaptic transmitter glutamate by the activity of plasma membrane glutamate transporters. And thirdly, there is the change in these astrocytes, that is a neurotransmitter release from the presynaptic terminals seems to activate a flux of calcium, and this calcium levels elevate in turn in the astrocytes, and they trigger the release of gliotransmitters in those networks where an astrocyte talks to other cells, both to neurons and to other astrocytes. And what are these gliotransmitters? There are three of them that have been discovered: D-serine, glutamate, and ATP. And all of these in turn have feedback modulatory effects on neurons. So let's have a look at them a little more closely. And what Philip Hayden and Michael Halasa, who was probably his fellow at the time, also hypothesized is that what you see here in this diagram is on the left hypoactive physiology, on the right hyperactive, and you have three different representations of astrocytes. You can see the astrocytic cells, and in the middle bar you can see calcium oscillations that are shown, and below that is the NMDA function in the postsynaptic neuron. So what they hypothesized is that depending on the physiologic activation of calcium oscillations in the astrocyte, whether it's physiological or hypoactive or hyperactive, the NMDA potential that is generated in neurons that are part of this tripartite synapse, they can vary. So it can be, its amplitude can vary, as you can see, small amplitude in hypoactive, normal amplitude in physiological, and a higher amplitude in hyperactive. And which means that the astrocytes have an important role to play in the modulation of neuronal activity. And they also demonstrated through their work over a period of years that the three glial transmitters secreted by astrocytes, glutamate, ATP, and D-serine, have differing anatomical effects on the receptors on which they act, which is shown here in highlighted in yellow as the target. You have NMDA receptors, you have kainate receptors, you have the P2X receptor, you have the adenosine receptor, and depending on where in the brain um, these glial cells function, they can have differing activity and differing function, and we will discuss that in further slides to come. So essentially, if we were to understand that what are the three key functional roles of astrocytes, then one, the key functional role shown here is buffering of potassium in the synaptic space. And this buffering occurs because in the astrocyte, there is what is shown here as KIR 4.1. That stands for potassium inward rectifying channel. So this is a channel that regulates and takes up potassium when a neurotransmission occurs, and it disallows the neural transmission from becoming haywire and from doing anything that it is not supposed to do. The second function of the astrocyte is regulation of aquaporin 4. In my third lecture last Saturday, you heard about how aquaporin 4 has an important role to play in the lymphatic function. And we also believe that aquaporin 4 is the key regulator in the astrocyte that is responsible for water balance across synaptic membranes in the syncytium and the ensemble of neurons and astrocytes. And the third function shown here in red is the clearance of glutamate from the synaptic space. We know that glutamate is an excitatory neurotransmitter secreted by neurons. It is shown here in the diagram on the left. And these may continue to excite the postsynaptic receptors if they are not mopped up by the astrocyte. So the astrocyte has a glutamate transporter 1. It's also known as excitatory amino acid transporter 2. And what it does is to mop up glutamate. And this is a fine balance. So every time one neuron talks to another, there is a modulatory astrocyte that is fine tuning this conversation and making sure that this conversation is streamlined and harmonious and not in disarray, producing an activity that it doesn't need to do. He also demonstrated in the next few years that the astrocytes have a very important function. The astrocytes are uh, cells which also keep neurons replenished of energy by a complex process. So what is it? What he demonstrated is that astrocytes take up glucose from neurovascularly coupled vessels. This glucose is then metabolized by the astrocyte to lactate, and thus the astrocyte derives energy for itself. It then shuttles the lactate from the astrocyte to the neuron via a monocarboxylate transporter called MCT. And in the neuron, this lactate is then converted to pyruvate by an aerobic energy source. And by an aerobic process, it gets converted to pyruvate. And this source of lactate has shown to be pivotal for various neuronal functions that occur at night. And researchers label this system as the astrocyte neuron lactate shuttle. So for a period of years, this was thought to be controversial. Now it is accepted that this is absolutely real and it exists in most of the neural networks that are relevant for neuronal function. And this is again seminal work done by Philip Hayden and colleagues and replicated in other labs. And this diagram shows you how this transpires. So you have this capillary here on the right, and you have a glucose transporter that takes up glucose, and astrocyte takes up with the glucose transporter, it takes up glucose, take it within the astrocyte, it converts this glucose to lactate via intermediary of pyruvate, and this lactate shown here on the left of the astrocyte monocarboxylate transporter, it transports this lactate to the neuron, and at the same time there is an ongoing metabolism of ATP within the astrocyte, which generates um, from glutamate, it generates glutamine. It is shown here, GLU to GLN, that's a conversion of glutamate to glutamine via glutamine synthetase. And how does the astrocyte do that? It takes up glutamate from the synaptic cleft via the excitatory amino acid transporter. It converts the glutamate to glutamine, and then the glutamine is shuttled back to the neuron, and that glutamine is then converted into glutamate by the neuron for um, uh, neurotransmission. So what it's showing us is that the astrocyte neuron lactate shuttle is the energy source of the neuron. So the neuron does a lot of work in the day, and when we are sleeping at night, the astrocyte neuron lactate shuttle replenishes the energy source of the neurons to have it ready for functioning the next morning. Now that's a really astounding concept. So this diagram is a representation of all these complex functions of the astrocyte represented together. So what you have here is the astrocyte in the center. You have the neuron on the left, the presynaptic and the postsynaptic terminal, and on the right you have the capillary. And you can see the foot process of the astrocyte, which abuts this capillary. And what is shown here is the neurovascular coupling, and we will come to that in the next couple of slides, that the astrocytic foot process has its own biochemical mechanism which regulates this coupling by allowing the dilatation and constriction of vessels to occur when there is ongoing neuronal activity. We all know that we do bold imaging or functional imaging on the MRI. And what is that functional imaging? It is change in hemoglobin and oxygenation of local brain tissue when we do work, and that is reflected on the MRI. Now that change is a vascular change. So whenever these synapses do additional activity, which is functional, it changes the vasculature, and that vascular change occurs because of this neurovascular coupling of the astrocyte on the blood vessels shown here. And this cascade of events, the excitatory amino acid transporter to mop up glutamate, the invert rectifying 4.1 current to mop up potassium, and glutamine synthetase within the astrocyte to generate glutamine, which shuttles back to the neuron. I mean, it's completely fascinating. And shown on the top is the lactate shuttle. So the neuron is actually doing a whole amount of work while we are sleeping at night. And it could also explain the function of aquaporin 4 is not just within the realm of glymphatic regulation, that is a major function, but it also functions in maintaining a water balance in the cerebral tissue by harmonizing how water content flows in between the interstitium of the brain, the neuron, and the astrocytic regulatory processes. And these, as we can imagine, can get altered if an astrocyte is sick. When does the astrocyte become sick? It can become sick when there's a head injury, when there is hepatic encephalopathy. We know that hepatic encephalopathy pathologically is associated with type 2 astrocytic swelling. When the astrocyte swells, there is water retention. Hyponatremia does the same. When there is a loss of neurovascular coupling, for example, with accelerated hypertension, it produces reversible posterior leukoencephalopathy. We all know that this is a kind of reversible focal cerebral edema, and now we can recognize the mechanistic processes in an intricate manner that reflect how these changes occur at the bedside. So the astrocyte is a quiet and industrious renovator in sleep. It determines unidirectional flow of CSF during glymphatic activation. We learned about that in the last lecture, and we know that how polarization of aquaporin 4 is responsible for optimized glymphatic function. We know that sleep homeostasis is driven by adenosine accumulation. So the homeostatic drive to sleep in the evenings is dependent on the accumulation of adenosine, and this is dependent on astrocytic production And we'll talk about that in the next few slides to come. Philip Hayden again was the first to demonstrate that the source of adenosine is not necessarily neuron, but the source of adenosine is actually the astrocyte. And we'll come to that in a minute. Astrocytes also regulate neuronal slow oscillations in cortical pyramidal cells, producing the slow wave activity that represents sleep. They take up glucose, metabolize it to lactate, and shuttle it to neurons via the monocarboxylate transporter, thus energizing the neuron for daytime function. And all this is pivotal for sleep-associated memory consolidation and seizure suppression. And we'll come to that in a minute and understand how it is intricately woven. So this paper by Brian McVicar shows the methodologies at a biochemical level within the astrocyte that also regulates neurovascular coupling. So it was believed that whenever there is an increase in synaptic activity, there is nitric oxide which is produced by calcium currents within the neuron, and this nitric oxide then gets converted to cyclic GMP and it dilates the vessel. We now know that there is an additional mechanism driven by astrocytes. Astrocytes produce from arachidonic acid via the prostaglandin pathway two different metabolites, icosa tetranoic acid shown as EET, and there is a hydroxyecanoic tetraacid shown as HETE. And these two have differing uh, effects on the blood vessel. It can either constrict or dilate. And the current conceptualization is that in this complex cascade, there are three different biochemical effects. So the astrocyte is in a position to either constrict or to dilate the blood vessel by producing from arachidonic acid via complex prostaglandin pathway products that can either dilate or constrict it. And this works in close conjunction with the effects of adenosine, which we know can have a direct effect on smooth muscle cells. And adenosine itself is a vasodilator because it alters smooth muscle calcium currents. So all this could be relevant in various disease processes. And the role of adenosine has become a very significant fifth regulated function of astrocytes. So what is shown here is the complex relationship of adenosine in the synaptic clefts. We know that adenosine is a byproduct of ATP metabolism. By nucleosidases work on ATP converted to ADP. The end product is adenosine. So what you have here on the right is an astrocyte, and the astrocyte on its membrane has what is called the equilibrative nucleoside transporter. The equilibrative nucleoside transporter takes up adenosine from the cleft and it converts it to AMP and ATP, and it regulates the amount of adenosine that is available. So you can say that the astrocyte functions as a policeman of the amount of adenosine that accumulates in the basal forebrain as a part of the homeostatic drive. And we know that one can imagine that because of all these complex functions, how astrocytes could be very relevant in its um, anatomical and microarchitectural contributions to a fine balance in the synaptic cleft that regulates depolarization shifts, which we know maintain normal neuronal chatter and disallow hyperexcitability to occur, which characterizes seizures. And in this review that was published in Epilepsy Currents, they is very aptly titled "Astrocytes: The Stars of a Sacred Disease." The sacred disease is what the Greeks thought was sacred, is epilepsy, and we know that potassium buffering, glucose regulation, buffering of glutamate, and adenosine homeostasis and gap junctional coupling are all relevant to maintain a homeostasis of electrical currents in the brain, and all of them could get deranged in epilepsy. So let's look at some of the indirect evidences that we have that suggest that such could be the case in humans who have seizures. So during neuronal hyperactivity, potassium is released by active neurons, and this is primarily taken up by astrocytes. So any impairment of the uptake mechanism by the inward rectifying channels 4.1 is if this changes, it could affect seizure control. And this has been demonstrated in hippocampi. Even if you expose them to millimolar or submillimolar increases in potassium in models of epilepsy, it enhances epileptic form activity. And these potassium inward rectifying channels are responsible for uptake of potassium, and downregulation of these channels occurs in injured or diseased CNS. There is some direct evidence of downregulation of potassium currents in sclerotic CA1 regions of the hippocampus. This has been shown in some studies. This was published in 2000. There is other data which show that astroglial potassium inward rectifying channels could be a contributory mechanism to contribute to hyperexcitability in epileptic tissue. How and why this occurs in say focal dysplasia is something that is being explored, and we don't know everything yet. The other mechanism by which epileptogenicity could be altered in the brain is the astroglial water regulatory mechanism. We know that brain tissue is exquisitely sensitive to osmolarity. Any size increase in the extracellular space can change that. If you decrease the extracellular space, it becomes hyperexcitable. If you enhance the extracellular volume with hyperosmolarity, then it attenuates epileptic form activity. And these experimental findings suggest that this could be an explanation for what happens in the human brain when there is hyponatremia of a critical degree, say below 121, and when we know that one of the common reasons for late onset seizures is hyponatremia, often driven by medications like diuretics. We also know that there is some indirect evidence that aquaporin 4 localization and expression gets altered in humans who are epileptic and who have undergone surgery. And this has been demonstrated in sclerotic hippocampi. Uh, it's been shown that there is mislocalization of aquaporin 4 in the epileptic hippocampus with a reduction in perivascular volume membrane expression. And researchers imply that a change in aquaporin 4 function anatomy in conjunction with inward rectifying current abnormalities result in the propensity of seizures in epileptic tissue. What about glutamate clearance? We have several lines of evidence which seem to show that. There are studies in mice which show that if you delete or antisense oligonucleotide mediated inhibition of this astroglial transporter called the excitatory amino acid transporter 2, it produces a greater propensity to have seizures. And it's been shown that this glutamate transporter immunoreactivity is decreased in the sclerotic hippocampus, which we know is a model for refractory seizures. The other mechanism is glutamine synthetase, and it's been shown that glutamine synthetase levels are variable if you compare sclerotic versus non-sclerotic hippocampi, and this could perhaps affect the uptake of glutamate into astrocytes and could be responsible for worsening seizures. So all these studies are indirect modes of analysis and scrutiny that imply that the astrocyte has an important regulatory function in, in health and in disease. And in disease, there could be multiple mechanisms that are dysregulated in the condition of epilepsy. And of course, this glutamate homeostasis by the astrocyte has now deeply entrenched in basic science literature, and this is supposed to be a very important function of the astrocyte. What about adenosine? What does adenosine actually do? We know that adenosine levels are elevated during seizure activity. Adenosine exerts a very powerful inhibitory effect on excitatory synaptic transmission, mainly through its interaction with presynaptic A1 adenosine receptors. Remember, there are A1, A2, and A3 different receptors, and we are trying to figure out how they have differing modulatory effects on seizures and sleep. Once released from neurons and astrocytes, ATP is converted to adenosine monophosphate and then into adenosine by extracellular nucleotidases. This reuptake of adenosine occurs, as I showed you in that diagram, through equilibrative nucleoside transporter, and phosphorylation of by the astrocyte-specific adenosine kinase breaks down adenosine and clears excess adenosine from the extracellular space. So adenosine is like the janitor I mean, the astrocyte is like the janitor that clears the adenosine and keeps the synaptic cleft clean and makes sure that an adequate amount of adenosine is present in the synaptic cleft, both for inhibition of seizures and for induction of sleep. And this is relevant in generation of seizures. There are some indirect evidences which show that in brain-resistant seizures and injury, this can change. Adenosine kinase levels are overexpressed with astrogliosis, and spontaneous seizures occur in rodent models when adenosine um, kinase levels are overexpressed. And several lines of evidence confirm that this can occur in the epileptic brain. We also know that adenosine buildup is responsible for the homeostatic sleep drive on the flip-flop switch of the VLPO neurons that secrete gamma and galanin. We discussed that in the first lecture in this series. And it's very clear that the astrocyte is an important contributor to all these functions. So here is a summary of all the things that the astrocyte can do, which is anticonvulsant. That's shown on the left. Via the inward rectifying potassium channels 4.1, it uptakes up potassium and buffers it, keeping the synaptic cleft clean and decreasing epileptogenicity. It takes up glutamate, it regulates water, it converts glutamate to glutamine and keeps this balance. It changes ATP to adenosine, and adenosine has anticonvulsant effects. It maintains gap junction coupling. On the other side, if glutamate and D-serine release occurs, if there is an excessive amount of ATP in the cleft and there is an alteration in adenosine metabolism, um, and if there is a break in the blood-brain barrier due to TGF beta, then this break in the blood-brain barrier also increases seizure risk. And we'll come to that in a couple of minutes. So we know that there are multiple mechanisms wherein astrocyte function is relevant for patients with seizures. If you remember, we discussed this question with Dr. Lowenstein a couple of days ago. There is a group in Colombia that is working on these models to explain astroglial function in the pathophysiology of status epilepticus. And this diagram is again a representation of those functions that we discussed: clearance of adenosine, clearance of glutamate in the synaptic cleft, and the regulation of blood-brain barrier by neurovascular coupling as shown on the left. So the hypothesis is that a disruption in blood-brain barrier could allow albumin to pass from the capillary to the brain parenchyma, and albumin has a deleterious effect on the expression of aquaporin 4, and it produces cerebral edema. In addition, it dysregulates the inward rectifying potassium current. Astrocytes swell up, there is intracellular calcium, it alters an aberrant amount of gliotransmission, so the amount of ATP goes up, adenosine decreases, and because of all these mechanisms, there is perpetuation of seizure. And this concept that the blood-brain barrier, perhaps in recurring seizures, is disrupted and could be a mechanism that could be addressed in therapy has taken the world of epilepsy by storm. And in this paper by Jan Gorter, it's very aptly titled "The Roof is Leaking and a Storm is Raging." So when you have recurring seizures, the blood-brain barrier is disrupted, and perhaps this could be one of the features that need addressing in an attempt to understand this. So the purists first contemplated that what is the actual truth? Is this disruption of blood-brain barrier a cause? Is it a consequence, or is it both? And Wolfgang Loescher, whom many of you may have met in the international epilepsy meetings where he speaks about refractory seizures and their mechanisms, has published this paper just about a few months ago, contemplating that the next frontier in the science to regulate refractory seizures should perhaps also be looking at neurovascular coupling, because this whole process of neuronal modulation by astrocytes is closely linked to the efficiency of neurovascular coupling, and this could be a window to understanding recurring seizures better. And in various very illustrative and insightful publications in epilepsy currents, various experts talk about how a breach in the blood-brain barrier in animal models could result in a disruption of the cation chloride cotransporter channels, producing an alteration in inhibitory currents and producing seizures. So we know for sure that the regulatory functions of astrocytes have a remarkable effect on the metabolism and balance of potassium, glutamate, and adenosine. And we discussed this diagram earlier. This diagram is also relevant in the adenosine regulation of sleep. We know that a buildup of adenosine in the basal forebrain is responsible for the homeostatic drive to sleep. And we now know that there are three types of adenosine receptors, A1, A2, and A3. And current science is trying to figure out the distribution of these in the basal forebrain. And based on the type of receptor and the distribution, it is postulated that adenosine may have differing physiological functions in the human brain. We also have some indirect evidence which shows that if the human brain is sleep deprived, the A1 adenosine receptor binding is upregulated so that it increases the homeostatic drive to sleep. And this has been shown in a PET study shown here. And those who are interested can look up this paper by David Elmenhorst. So you will agree with me that Philip Hayden's conceptualization of the tripartite synapse between the astrocyte and the neuron has changed the way we think of brain physiology. And the current understanding is not only do glial cells have an important physiologically regulatory function, but they also control synaptogenesis and synaptic pruning. And in the normal brain, it's shown here on the left, the researchers have identified various epitopes in the surface membrane of astrocytes like thrombospondins, heavin, spark, and glypican 4 and 6, and chondrin-like 1. These are all receptors which have been detected in animal studies, and it's believed that these receptors are responsible by their regulatory function in producing synaptogenesis, so that when the brain is sleeping, which we learned that after doing a learning task, when we sleep, synapses increase. These synapses, synaptogenesis is regulated by astrocytes. We also know that synaptic pruning that occurs, we talked about the synaptic homeostatic hypothesis in the first lecture, and we know that inadvertent memories are deleted and the advertent ones are consolidated when we sleep. The deletion and pruning seems to occur with MGF10 and MERTK. So what this tells you is that glial cells have an important regulatory function. They regulate physiology, they produce synapses, they remove synapses, and when diseased, they can produce brain injury. And what is shown in the right is diseased brain in various models of disease, which includes Alzheimer's disease and Parkinson's disease and ALS, wherein we know that different complement and interleukin and tumor necrosis factor alpha related change in activity of glial cells produces neuronal injury. So astrocytes do contribute to synaptogenesis. Misregulation of glial function can lead to synapse loss. We know that astrocytes and microglia secrete synaptogenic factors, thrombospondin, heavin, spark, glypican 4 and 6, and chondrin-like 1 from the astrocyte. BDNF and interleukin 10 from the microglia also regulates synapse function. Synapse pruning can occur with a different mechanism, MGF10 and MERTK phagocytic pathways for the astrocyte, and through a complement cascade in microglia. And there was some discussion about how this can occur even in autoimmune diseases like myasthenia gravis in some of the conversations on the MAN group. Astrocytes can also regulate microglial mediated synapse elimination by secreting transforming growth factor beta and interleukin 33. So we know that sleep fragmentation upregulates microglial transcripts. And one can imagine that if these microglial transcripts get upregulated and they secrete a larger amount of this TGF beta and interleukin 33, it could result in synapse loss and loss of function. Voluntary exercise, on the other end, we discussed this in various parts of my earlier lectures, seems to have the opposite effect, and it's important for neurogenesis. There is an attempt now to understand these benefits of voluntary exercise in the light of our understanding of these various mechanistic modalities of brain health. So what is shown here is that exercise can increase astrocyte size, it can improve astrocytic aquaporin for localization and expression, and can increase astrocytic transporter levels. In an indirect fashion, astrocytes can also modulate synaptic growth during and after exercise by increasing glypican, thrombospondin, heparin, and spark and BDNF levels as shown on the left here. So essentially, the belief is that glial cells have a major contribution to make both in memory consolidation, memory function, and cerebral regulatory growth. And many of these mechanisms can become aberrant in sleep disorders and Alzheimer's disease. And as we've learned in the previous lectures, all of these are closely intertwined, and it's an exciting time to understand how this actually occurs. What about some more functions of astrocytes? In an astounding um, bunch of investigative attempts, it's been found that hypothalamic astrocytes have some unique functions. We know that the hypothalamus is a regulator of sleep, thirst, hunger, circadian rhythms, body temperature. We discussed that in lecture 2. And it possesses a unique biofeedback mechanism due to fenestrated capillaries in the ventromedial region. Now, hypothalamic astrocytes astoundingly express receptors for hormones. They express receptors for leptin, ghrelin, insulin-like growth factor 1, glucagon-like peptide 1, thyroid hormone, and insulin. And leptin and ghrelin balance is sensed in sleep by astrocytes in the hypothalamus, and it is regulated by adenosine-mediated inhibition of this agoutin-related peptide producing neurons in the arcuate nucleus. So what it's telling us is that astrocytes seem to have a very important neural endocrine feedback loop regulatory effect. And this has been shown in various studies. This is a Korean study which demonstrates that leptin signaling in astrocytes regulates neuronal functions for feeding and for satiety. And it's not difficult to understand that they perhaps are also the emerging stars of energy homeostasis. And in this paper that was published in Cell Stress in 2018, it is believed that astrocytes are morphologically diverse in males and females, and they could have a role in this sexual dimorphism, could have a role in regulating the endocrine functions in women. And this has become the focus for future research. For those who are interested, they should read up this paper by Simonetta Camandola. It's really quite an eye-opener. And in a very, very recent publication, Zhang and his colleagues have shown that they have demonstrated a mechanism that astrocyte process plasticity secretes various mediators which regulate blood glucose, blood pressure, and body weight. So what they are implying is that if one is hyperglycemic, if one is obese and one is over nutritionally endowed, then it could further alter the bioregulatory mechanisms that control blood glucose and fat stores and could worsen the chronic inflammatory state of diabetes. And lastly, we have some emerging evidence from Chiara Cirelli's group in Madison, Wisconsin Madison, which shows that almost all aspects of sleep that we discussed in the first 3 lectures, especially the oscillatory slow wave activity and its dynamic regulation in sleep, they are all amply regulated by astrocytes, and various genes which have effects on this astrocytic control are now being demonstrated. And we know that slow wave oscillations, again, in the forebrain, there is this D-snare experiment. D-snare is a membrane vesicular protein, and it is indigenous to astrocytes and not to neurons. So if you take away this domain, then slow wave oscillation activity changes in optogenetic models. And that's been shown that the slow wave oscillations in the characteristic sleep regulated phenomena in both animals and humans is regulated by astrocytes and is regulated by adenosine. And the current area of research is that every aspect of the EEG activity that we know, um, theta activity, delta activity, alpha activity, beta activity, and gamma activity, all of these are regulated by complex astrocytic function, and depending on where in the brain this electrical activity occurs, it is thought to have differing physiological effects. And for those who are interested, you can look up this publication, which demonstrates that these phenomenologies can be understood by a complex regulatory function of astrocytes. Now, many of you know that um, when Albert Einstein died in 1955, when he was cremated, his pathologist took away his brain and preserved it, and Thomas Harvey actually tried to give it back to his family later. And one of the reporters, Michael Paternity, wrote about this trip when he drove across America with the last vestiges of Einstein's brain. And it's published in a book called Driving Mr. Albert. And this is a story in itself. And Douglas Fields, who's done a lot of work on glial function, writes about this and says that Marion Diamond, who published a paper in 1985 saying that there was a change in the glial morphology in the parietal cortex of Einstein's brain. And this became very controversial because subsequent to 1985, from then to the early 2000s, about 7 or 8 different scientists argued against such derivations being made. But it led to the concept that perhaps there is a role for glial biology in learning and cognition. And Douglas Fields talks about it and says that this needs exploring. And in one model, a humanized chimeric mouse was created, wherein a large proportion of mouse astrocytes were replaced with human astrocytes. And the moment that happened in the petri dish, there was an increased synaptic plasticity and faster learning. So in models, we are learning that astrocytes can have relevant modulatory effects on learning and cognition. And Douglas Fields has written a book called The Other Brain. Sudhir is a very big fan of Douglas Fields. I think he should invite Dr. Douglas Fields to talk to us about why it could be important for neuroscience enthusiasts to map this other brain, the other brain being glial cells. And I'm pretty sure that as time goes by, we learn more about it. Very recently, there's been a publication which supports a model in mice wherein there is a demonstration that astrocytes can regulate cognitive flexibility and neuronal oscillations. And we know that calcium binding occurs through this protein, which is unique and which has been isolated now. So if we are at the cusp of dramatic amount of knowledge on how astrocytes function, and this should excite us as neurologists so that we can study this better and perhaps we can share this understanding with our students and colleagues. And there is another school that believes that I discussed with you the energy homeostatic roles of astrocytes in the hypothalamus. The hypothalamus also has tan sites, and it's believed that these tan sites are perhaps a bridge between ependymal cells and astrocytes, and they are a morphologically altered form of astrocytes. They have receptors for leptin-2, and they are responsible for um, uh, taking up signaling from the bloodstream and the CSF, taking up signaling from leptin and ghrelin, which we know occur in a fine balance to regulate feeding and satiety cues for animals and humans. And I think this will be explored further in time to come. We're also discovering some new glial cells like the NG2 cells. It's been discovered by the Japanese group a few years ago. And they were able to show that these NG2 cells have immunomodulatory and function in hippocampal neurons. And the point is that we are still at the cusp of a huge revolution in understanding how glia contribute to almost every aspect of neuronal function. I'm going to stop here, and I hope that with this diatribe, I've managed to increase your enthusiasm in studying some aspects of science, because if we don't know this, we will not understand our patients the way we should. Thank you very much for your listening.
Thank you, Joy. And I think you've really gone into the depth of the whole process of how astrocytes are very important, particularly for mediation in sleep. We have more than 100 participants, and we have questions. Are you ready and okay to take the questions?
Yeah, I'm good. I'm good. I'm with
Okay.
you.
So there's one question which is very interesting. What makes glial cells prone for neoplastic transformation? Is it metabolic peculiarity, being active in activity, rest, and sleep? So basically,
Yeah.
probably this attendee is aware of the fact that sleep has a huge role to play in the occurrence of malignancy, and we know the lack of sleep is associated with certain malignancies like breast cancer. It ups breast cancer to almost 30 times as per the research says. So basically, do the glia have anything to do with this kind of regulation in sleep?
So yeah, there. In fact, I came across 2 or 3 publications, and if I don't know who is asking this question, but if they message me individually, I can
I
share with
can't.
them. Um,
There is no anon. It's like
yeah,
SMA 700FD. So
yeah,
there's no name there.
yeah,
So
yeah. So the thing is that if you search this, there is some emerging data
hello.
which shows that we know that astrocytomas behave biologically differently from neuronal tumors. We know that glioblastomas are derived from a precursor cell to astrocytes in the glial lineage, and a lot of work is being done to understand upregulation of oncogenes. There are studies which demonstrate for other tumors, like the Norwegian study, which shown that if you are sleep dysfunctional and circadian rhythm dysfunctional, then oncogenes get upregulated. Matthew Walker's group has shown in some of their studies that clearly oncogene upregulation and dysfunctional sleep are related to each other. I am not aware of brain tumors in specifically for brain tumors how this occurs. We know that from the Norwegian study that is relevant for lung cancer and breast cancer in men and women. And I'm sure that with a better understanding, we will know more about this. I personally haven't read a great deal about glial tumors and astrocytic mechanisms.
Yeah, thanks, Joy. And I just have a question. Like the most favorite substance which we consume, or we are kind of softly addicted to, is caffeine. And we know
Right.
that caffeine works in keeping us awake, at least,
Yes.
by working on the adenosine receptors.
Yes.
So what it does is it blocks the adenosine receptors by
Correct.
making the adenosine receptors believe that it is adenosine. So it
Correct.
mimics adenosine and blocks them. So
Yes.
we know when adenosine blocks to the adenosine receptors, it produces sleep. I mean, it is sleep promoting. So when we take caffeine, it's blocking the action of adenosine on the adenosine receptors, and obviously the person is not asleep. Is there a difference in the way caffeine works in different, glial cells or neurons, or is there any difference of mechanism, or does it have a larger? And then there's some of these people who say that they will consume tons of caffeine and still not sleep. I mean, still sleep. So
Yeah.
they
So
take caffeine just 1 hour or half an hour before bedtime, and they still sleep. So
right.
it doesn't make a difference to them. So it's very peculiar that some of
Yes.
us are awake with caffeine and some of us fall asleep,
Yes.
despite how
So
much we take.
that's a very interesting question. I've thought about this at length, and some of my students who are probably listening in today will remember the journal club that we did on caffeine, which was done by Swati Sharma, my resident, and we addressed this, looked at literature, and there are a few thoughts I have on this subject. One, it appears that adenosine seems to have in different parts of the brain the ability to bind to different subsets of adenosine receptors. We know that it's been shown that A1, A2, and A3, and A2 has subtypes A and B. So there are two subtypes of A2 as well. So it's not very clear as to what are the different functions between these. Are these receptor subtypes different? Is the distribution in the brain different? This is probably work in progress. But there are a few interesting observations which came through. Many of you, I think Roop might be listening in today. He might remember the last president of the American Epilepsy Society, Barbara Yost. She gave a lecture at the Baltimore AES last year, and she shared a lot of data from Philippa Caroli from Australia, where neuropathy data in patients with epilepsy was being shown, and they were showing the occurrence of interictal spikes in these implanted patients. And one peculiar finding that came about is that when patients drank coffee in their diaries, the density of interictal spikes, they didn't have seizures, but the density of spikes increased. And uniformly across that 5 and 7 year of observation, when they analyzed the seizure diaries of these neuroplace implanted patients, clearly there seemed to be a relationship between consumption of coffee and increase in interictal spikes on their mapped on their electrodes implanted in the brain. So Barbara Yost jokingly said that, should we tell our epilepsy patients to stop drinking coffee? She said this on the dais. Now, if you look at what we know, it's fairly clear that if we do not allow adenosine balance to occur, we know that adenosine has anticonvulsant effects. And this is perhaps an indirect demonstration that by consumption of coffee in amounts maybe larger than usual, one could be at risk of tipping the balance, so to speak, in the regulatory mechanisms that control seizures in our patients. And I, for one, at least understood from her conversation and whatever little reading I've done, is that perhaps it may make sense for patients not to drink coffee in very large amounts. Maybe 1 cup of coffee per day is fine. The other issue is that Matthew Walker, in a lot of the work that's been done and published in his book, Why We Sleep and Dream, clearly says that the half-life of caffeine is at least 6 to 8 hours, which means if you have coffee by 3 o'clock in the afternoon, it stays in your system by the time you go to sleep at 10:30. And he also makes a point of saying that consumption of coffee late in the evening, you may still be able to sleep by many people who claim that they sleep, but their depth of sleep in... How much is remaining? I need the Wi-Fi. 5 minutes. The depth of sleep is shallow, and therefore a larger amount is spent in N2 sleep, not in N3 sleep in coffee drinkers. This is again work in progress, but he's written about it. So that could answer some of the questions that you asked.
Yeah. I think that's all we have. And the person has identified the first question. The cancer one is Dr. Krishna Prasad. So we
Right.
have an identity. Thank you for asking that question. I think Dr. Sudhir, is he around?
Yeah.
I think we can, kind of wind up. And thank you
Yes.
so much, Joy, for talking to us about this topic, which we normally don't read, or we don't, have the patience to even think of reading. So really, you brought us, on the platform and mechanisms and neurobiology and the basics of together.
Right. Thank you so much.
Thank you.
Thank you. It was a pleasure. Thanks. Tell Mama it's done.